secondary lymphoid organs. So what do we actually mean by this secondary lymphoid organs? Secondary lymphoid organs are the organs which are actually responsible for the process of activation of the B and T lymphocytes, right? Secondary lymphoid organs are responsible for activation of the B and T lymphocytes. So two major types of secondary lymphoid organs what we'll be taking up. The first one what we'll be talking about is the spleen. Right, so this is the first primary secondary lymphoid organ what we are actually taking about, which is called as spleen. So we are all very much familiar with this particular structure called as spleen. We have come across a lot of times about this particular structure also. So what exactly is this spleen? So it is a large bean shaped structure which is actually present in our body. Regarding the function of spleen, we know one major function of spleen is they start acting as the graveyard of RBCs. RBCs after they die, right, after they undergo the process of lysis or death, right, so they usually get or they usually are stored up in this particular spleen. In the spleen, the RBCs break down into different parts. So all that we have already studied. So we are not going to get into the details of it, right? So spleen is actually considered as the graveyard of RBC. Now, along with that, spleen also has one more work to do pertaining to the immune system present in our body. Right. So what does the spleen actually contain? The spleen actually contains something called as the phagocytes as well as mature lymphocytes. That is both the T as well as the B lymphocytes, right? So this spleen usually consists of the phagocytic cells, such as macrophages, mostly the macrophages, and along with that, they also contain these mature lymphocytes, both the T lymphocytes as well as the B lymphocytes. That is what this spleen has. Along with this, the dead RBC is also being present here, right? Now, what does the spleen actually do? Spleen starts acting as filters. Right, so they usually act as filters. So we are talking about the function of spleen pertaining to the immune system. Normal function of spleen, we know it starts acting as a graveyard of RBC. But here we are talking about the function of spleen pertaining to the immune system that is present in our body. So spleen acts as filters. Filters for what? Filters for the blood-borne antigens. Right, so they start acting as filters for blood-borne antigen. So what does this mean? Whenever an antigen enters inside our body, right? So whenever an antigen enters inside our body, that particular antigen usually enters into the bloodstream. It enters into the blood circulation. So as it enters through the blood circulation and passes through different parts of the body, they usually come and get trapped here in the spleen. So what the spleen does is, spleen will start filtering all the antigens that are actually present in the blood, that is present in the blood circulatory system. So all the antigens present in the blood circulatory system or the blood circulation are trapped by the spleen. That's the reason why we call them as filters. Filters for what? Filters for blood-borne antigen. So whatever antigen enters into the bloodstream or into the blood circulation, the spleen will start trapping those particular antigens. Now, once the spleen has trapped the antigen, what would happen? Let's just suppose that this is the organ, which is the spleen, right? Let's just imagine that this is the spleen. Right? So the blood has passed through the spleen, and we also saw that in the spleen we have phagocytes and mature lymphocytes. So here let us write down that this is the phagocytes. So here we have the phagocytes, and along with that we have the B cells, and along with that there are these T cells which are being present. So the B and T cells, they are said to be matured B and T lymphocytes. So B lymphocytes after maturation, right, in the bone marrow, from the bone marrow they usually move into the spleen. Similarly, excuse me. Similarly, thymus, once they are formed in the bone marrow, they move into the thymus for the process of maturation. After maturation, from the thymus, they start moving into this particular organ called the spleen. So in the spleen, we have the phagocytes, we have the matured B cells and the matured T cells. Now, as the blood starts passing into the spleen, whatever antigens are present in the blood are being trapped here in this particular spleen. right? So all those antigens are being trapped in the spleen. That's the reason why we said spleen starts acting as a filters for blood-borne antigen. So any antigen present in the spleen, present in the blood circulation will be trapped here by the spleen. Now, in the spleen, antigens are present. In the spleen, phagocytes are there, B cells are there, T cells are also there, but they have not activated. So we have already studied that for the activation of either the B lymphocytes or the T lymphocytes, antigens are required. So matured B cells are there, matured T cells are there, but they are still in an inactive state. For their activation, antigen is required. So this antigen now has been trapped by the spleen. Antigens present in the blood has been trapped by the spleen. Now what would happen? This antigen will start, consider this to be the B lymphocyte, right? Consider this to be the B lymphocyte. On the B lymphocyte, we know that there are certain receptors which are being present. We call them as B cell receptors. So this antigen will go and bind to the B cell receptor. So once they bind to the B cell receptor, this B lymphocyte will now become activated. Then you know what exactly happens. All that story actually repeats. We are all familiar with that. Once B cell undergoes activation, this B cell will now undergo the process of differentiation. Once they undergo differentiation, memory cells and plasma cells are being formed, right? The memory cells will start remembering this pathogen or this particular antigen. And what does the plasma cells or the effector cells do? They would start synthesizing antibodies against the specific antigen. So once the antibodies are synthesized against the antigen, what does those antibodies do? They will now start targeting this particular pathogen. So how do they target? That also we have seen in the previous session when we were talking about humoral immunity. They either neutralize the antigen, they bring about agglutination, precipitation, immobilization if there is any bacteria. They start activating the phagocytes for undergoing the process of opsonization, and they also start activating the complement system. Therefore, whatever antigens are being here, those antigens are completely destroyed. Right. So that is the B cell activation. Talking about T cell activation, this also we have seen how the T cells will be activated, right? So if you could recollect what we saw with regards to cell mediated immunity, right? I told you that T cells will not be able to recognize the antigen as such. So before the T cell recognize the antigen, what should happen? The APCs, the antigen presenting cells, has to process the antigen. Once they process the antigen, that processed antigen has to be presented to the T cells. Only then the T cells will get activated. Once they get activated, they will get differentiated into T helper cells and T cytotoxic cells, right? So that's what is happening. Let us consider this phagocyte here to be a macrophage, one of the APCs, right? As and when an antigen enters into the body, this macrophage will start identifying this antigen. Once they identify the antigen, what does the macrophages do? They would undergo the process of phagocytosis. They would process the antigen. Once the antigens are being processed, we know that on the surface of macrophages, there are certain receptors present which are called as major histocompatibility complex class 1 and class 2. If class 2 is present, if antigens bind to class 2 MHC molecule, this antigen will be presented to T helper cell, right? The T helper cell will now undergo activation. Upon activation, what does T helper cells do? We are very much familiar with that. T helper cells would now in turn go and activate B cell, and at the same time, it would go and activate T cytotoxic cells also. If the processed antigen is present on class 1 MHC molecule, then what would happen? They would start presenting the processed antigen to T cytotoxic cells, upon which the T cytotoxic cell will undergo the process of activation. Once they undergo activation, they would start releasing perforins, granulozymes, and bring about complete destruction of the antigen. Right. So, secondary lymphoid organ, the spleen, right, is the site where there is activation of B lymphocytes and T lymphocytes happening. So that's the reason why we have called it as the secondary lymphoid organs, the site where there is activation of B cells and T cells taking place. We can also put it this way also, the site where the antigens interact with the lymphocytes are called as the secondary lymphoid organ. We can put it this way also, the site or the organs where the antigens interact with the lymphocytes, either the B lymphocytes or the T lymphocytes. We start calling them as the secondary lymphoid organs. So this is how the process of activation usually takes. Actually speaking, it is a very complex process. Just for your brief understanding, so I just made it this way. Hope it is clear. So this is how the process of activation of the B lymphocytes and T lymphocytes takes place in the structure called as spleen. So things what you need to remember: spleen, along with its function, which is the graveyard of RBC, it also is responsible for filtering the blood-borne antigens. So whatever antigens enter inside the blood, so all those antigens present in the blood are being trapped by the spleen. And we know that in the spleen, there are these phagocytes as well as the T and B lymphocytes also. Right? So they are waiting to get activated. So once the antigens are being trapped by the spleen, right? So once antigens are being trapped by the spleen, those antigens will now bring about the activation of either the T lymphocytes or B lymphocytes, such that these activated T and B lymphocytes will now bring about the complete destruction of the antigen that has entered inside the body. Right? So this is what we have with the first secondary lymph node organ, which is the spleen. Right. I hope this is clear. Right. Now let's get into the second lymph node organ, which is the lymph node. Secondary lymph node organ. The second secondary lymph node organ, which is the lymph node. The next one, what we'll be taking up is the lymph node. Right. So lymph node is again a small solid structures, right? So lymph nodes are nothing but small solid structures which are present in several points present in our body, starting from the face till the legs, right? We have a lot of lymph nodes which are very small structures, solid structures which are present throughout the body. Right, so they are actually the major constituent of the lymphatic system. I hope all are familiar with the lymphatic system, right? So we have two types of circulatory systems in our body. One is called the blood circulatory system, and the other one is called the lymphatic system. Right. So one of the major constituent of lymphatic system is this particular structure called as the lymph nodes, which are present in various points in our body. So starting from our face till the legs, we have a lot of lymph nodes that are being present. Now, what is the major role of this lymph node? These lymph nodes also function very much similar to that of spleen. So lymph nodes are also considered as filters. Right? So they are also considered as filters, right? So the lymph nodes are also considered as filters. Filters of what? Filters of lymph-borne antigens. Right? So spleen, we said, spleen is considered as the filters of what? Blood-borne antigen. Whereas here, in this case, in case of lymph node, they actually start acting as filters of lymph-borne antigen. So we know that there are two types of body fluids in our body, majorly. One is called the blood and the other one is called lymph. So lymphatic system, lymphatic circulatory system, other one is the blood circulatory system. Whatever antigens entering into the blood, they are all trapped by the spleen. Say suppose there is this antigen, rather than entering into the blood, they have entered into the lymphatic system. They have entered into the lymphatic circulatory system. So they are moving in the lymph. So whatever antigens have been entered into the lymphatic system, so those antigens present in the lymph are being trapped by these particular lymph nodes. So we said that in different points, we have these lymph nodes situated in our body or located in our body, right? So when these antigens present in the lymph travels through those respective lymph nodes, in those respective lymph nodes, all these antigens would be trapped. Now again, what does the lymph node consist of? Again, the same thing. The lymph node consists of phagocytes, right? They consist of phagocytes as well as the mature lymphocytes. The same thing what we have seen with regards to spleen, right? The lymph node also consists of phagocytes, the phagocytic cells, mostly the macrophages, and along with that, they also consist of these matured lymphocytes. So again, the same thing. In the lymph node, we have the phagocytes, the B lymphocytes, and the T lymphocytes. Once the lymph starts filtering those particular antigens, right? So those antigens will now be recognized by the B cells. Once the B cells recognize the antigen, again the same story continues. B cells undergo differentiation, they form the memory cells, the plasma cells forming the antibodies targeting against this antigen in the lymph. If it is the second way, T cell activation. What would happen? Since phagocytes are already there, APCs are already there, the APCs will first recognize the antigen. Once the APCs have recognized the antigen, then what would happen? The APCs would now start processing the antigens. Once they process the antigen based on the class 1 or class 2 MHC molecules, they would start presenting those particular processed antigens to the T cells, respective T cells, either it be T helper cells or T cytotoxic cells. So once they are presented there, the T cell activation happens. As a result of T cell activation, what would happen? The T cells, activated T cells would bring about the complete destruction of that particular antigen. Right. That is what is happening here with regards to the lymph nodes. Right. So spleen and lymph nodes, they are considered as the major secondary lymphoid organs present in our body. And these major secondary lymphoid organs are majorly responsible for bringing about the activation of the B cells and T cells. So if acquired immune system has to show its activation, if the acquired immune system has to show its activity, T cell and B cell activation is very, very important. And that T cell and B cell activation is usually done in this particular organs, what we call them as the secondary lymphoid organs. Just like how we said, primary lymphoid organs provides microenvironment for the maturation of T and B lymphocytes. Here also, the secondary lymphoid organs provides what? The microenvironment for the activation of the T and B lymphocytes. Right? So what exactly is happening here? Maturation is done by primary lymphoid organs, that is the bone marrow and the thymus. Activation is done by the secondary lymphoid organs, that is the lymph spleen as well as the lymph nodes. Right? So this is what we have with the lymphoid organs that are actually present in our body. The primary lymphoid organs, the secondary lymphoid organs. One of the major constituents of the immune system, more precisely speaking, one of the major constituents of the acquired immune system that is actually present in our body. Right? So this is what we have with the small topic called as lymphoid organs which are present in our body. Primary lymphoid organs, the secondary lymphoid organs. Primary lymphoid organs, we have bone marrow and the thymus. Secondary lymphoid organs, majorly we have spleen, the lymph node, and along with that we also have MALT, which is mucosal associated lymphoid tissue. Then we have something called as tonsils, and along with tonsils we also have something called as the Peyer's patch of the small intestine. Right? So this is what we have with the immune system, right? Immune system of the body, more precisely speaking, the lymphoid organs which are actually present in our body. Right? So with this, we are completely done with our topic called as the immune system, or the process called as immunity. Right? So before we wind up with this particular topic, we have one more small topic, the last topic to take up, which is called as immune disorders. Right? So let's take up with the last topic under immunity, which is called as the immune disorders. So I hope everyone are very clear with how the immune system actually functions in our body, what are the different organs of the immune systems, and what is the role of those particular organs present in our body, and how the immune cells targets the antigen, and how they will defend our body or provide resistance to our body against any pathogen or any disease causing organisms inside our body. Right. The last topic what we have under this chapter, under this particular topic, immunity, is the immune disorders. The immune disorders, right? So till now we have seen a lot of things, right? So till now we have seen a few things with regards to how the immune system of our body functions, right? So we know what the immune system does, what is the role of the immune system, and we know how the immune system of our body usually functions. Whenever there is any pathogen or any antigen or any foreign substance entering inside our body, right, what would happen? Our immune system would become activated. Once the immune system is activated, either it be innate immunity or acquired immunity, once the immune system gets activated, that immune system will start fighting against the pathogen or foreign particle that has entered into our body and protect our body from any sort of infectious diseases. That is the major role of the immune system. Now, when will our body, when will we be protected from the diseases? Whenever our immune system is functioning properly, right? So whenever our immune system is functioning properly, majority of the cases, right, so we are being protected from those particular diseases. But there are certain instances where there is improper functioning of the immune system, right? Sometimes there is some sort of improper functioning of the immune system. There may be several reasons for it. We'll talk about that also a few minutes later, right? So sometimes there is improper functioning of the immune system. So this improper functioning of the immune system would lead to something called as immune disorders. So these immune disorders would be so severe that sometimes it would lead to some sort of severe diseases in the individual, and sometimes it can even lead to the death of the person also. That severe the effect of immune disorders is, right? So whenever the immune system is functioning properly, everything will be well and good, right? Immune system will start protecting us from any sort of diseases or any pathogens or any infectious organisms. But sometimes, right? Sometimes the immune system will start functioning improperly. It does not function properly. This improper functioning of the immune system would lead to severe diseased conditions in the body, which can even lead to the death of a person also. Right. So those improper functioning of the immune system could be classified majorly into three categories. Right. The improper functioning of the immune systems could be majorly classified into three categories. So what are those three categories we'll take up? The first one is called as hypersensitivity. Hypersensitivity, or majorly we can also call it as allergy, but there is slight difference between hypersensitivity and allergy. We can consider it as allergy. The second type of immune disorders what we'll be talking about is called as autoimmunity. So this is the second type of immune disorder what we'll be talking about, which is called as autoimmunity. And the third type of immune disorder what we'll be taking up is called as immunodeficiency. Right? So all the improper functioning of the immune system, right? The improper functioning of the immune system could be classified into these three categories. First one, hypersensitivity, which we can also call it as allergy, right? Second one is autoimmunity, right? And the third one is immunodeficiency. I hope all are familiar with allergy, right? So there is nothing much to get into the topic called as allergy. We'll see in detail a few moments later. Talking about autoimmunity, what do we mean by autoimmunity? Auto means self. Immunity is providing defense against any particular thing. Immunity, right? Autoimmunity means what's happening here in this condition is our own immune system will start targeting our own body cells, right? Our own self cells. That sort of a disease we start calling it as autoimmunity. And the third one is immunodeficiency. From the name itself, we can make out there is decreased immune response. Immunodeficiency. Why there is a decreased immune response? Due to several factors. So we'll be talking about that also. Right. As long as our immune system is functioning well, the immune system would protect us from any sort of infection. But sometimes the immune system does not function properly. At that point of time, it would lead to this condition called as immune disorder. So three major types of immune disorders what we have: hypersensitivity, also called as allergy. Second one, autoimmunity, and third one is immunodeficiency. Right. So let's take up each one in little brief. Right. Let's begin with the first one, which is hypersensitivity or allergy. Let's take up with the first one. Right. So what do we mean by this hypersensitivity or allergy? So what exactly does this hypersensitivity or allergy mean? Right. So what do we mean by this hypersensitivity or allergy? First, just take down the definition, then we'll get into what does it mean. Right. So hypersensitivity could be defined as the exaggerated or inappropriate right? The exaggerated or inappropriate immune response, right? So the exaggerated or inappropriate immune response, right? Exaggerated or inappropriate immune response against certain pathogens or certain antigens, against certain antigens present in the environment. Present in the environment. This is how we actually environmental, MENT, right? So this is how we actually define the term hypersensitivity or allergy. So what does this hypersensitivity or allergy mean? So it is defined as the exaggerated immune response, right? The exaggerated or inappropriate immune response against certain antigens that are present in the environment. So what do we mean by exaggerated? Exaggerated is nothing but high efficiency is said to be very high response, right? Very high response, right? Inappropriate is so much of immune response is actually not required, right? Even though such very high immune response is not required, right? The immune system will start functioning that way. That we start calling as exaggerated or inappropriate immune response against what? Against certain antigens that are present in the environment, right? So what exactly does this hypersensitivity mean? Hypersensitivity could be defined as the inappropriate or exaggerated immune response against a particular antigen, right? So there is an antigen entering into the body, right? Normal immune response would be sufficient to bring about the destruction of that antibody. But what exactly is happening? There is very high force of immune response seen. Due to that, what exactly is happening is, along with the antigen, some of our body cells are also getting destroyed. Let me give you a simple example here. Say there is one antigen entering into the body, right? Just a single antigen has entered into the body. So to target this single antigen, say 10 antibodies would be sufficient. Just an example, right? Nothing, don't take it, think, just a simple example here. 10 antibodies would be sufficient to target this antigen, right? In general, normally, let us taking as an example wise, if there is one antigen entering into the body, 10 antigen would be sufficient to bring about the complete destruction of this. But here in hypersensitivity, what happened is, rather than 10 antibodies, right, 100 antibodies are being released, right? 100 antibodies are being released. Now, what does this do? 10 antibodies will go and start targeting one antigen. Remaining 90 antibodies, whatever are there, they would now start targeting the self cells that are present in our body. That condition we start calling it as hypersensitivity. This is what we mean by exaggerated immune response. High intensity immune response, we can actually call it as, right? So the intensity of the immune response is said to be very high. Actually speaking, such high intensity is not required. 10 antibodies would be sufficient to destroy this one antigen, but 100 antibodies are being released, such that along with the antigen, some of the self cells are also being destroyed in this particular process. That condition, we start calling it as hypersensitivity. Hyper is nothing but what? More. Hyper means what? Sensitivity, sensitive to something. Actually speaking, in this hypersensitivity, some individuals are sensitive to a few things, where for others it would be absolutely normal. We'll talk about that also. Right. Okay. So this is what we mean by this definition of hypersensitivity. Now, the substances which bring about this allergy, we start calling them as allergens. Right. So what do we mean by allergens? Allergens are nothing but substances which cause allergy. We start calling them as allergens. Right. So substances which cause allergy, we start calling them as allergens. So what examples can we give for allergens here? So the examples what we can give is dust, right? So dust is one of the allergens. Getting back to this term hypersensitivity. Hyper means, I told you, it is more. Sensitivity is some people are said to be more sensitive to a few things than the others. Let us take this example only, dust. Some people are said to be very highly allergic to dust. Say two people are walking on the same road where there is too much of dust. One of them, they start sneezing whenever they come across that dust, whereas the next person remains absolutely normal. So what do we mean? This person who starts sneezing is said to be hypersensitive to the dust, whereas the other one is normal. Other one is not hypersensitive to the dust. That's what we mean by this hypersensitivity. Allergy is again the same thing, exaggerated or inappropriate immune response against these particular antigens. So one of the examples of allergens what we can have is dust. Along with dust, some of them are allergic to pollen grains, right? Pollen grains. Some of them are allergic to animal dander, right? Some of them are allergic to some sort of fruits, right? Some of them are allergic to some sort of vegetables. Some of them are allergic to some sort of drugs, that is medicines such as penicillin, all that. So all these fall under the category of allergens, right? So the substances which bring about this sort of allergy or hypersensitivity, we start calling them as allergens. So these are usually seen, most of these allergens are usually seen in the environment, right? Such as the dust, the pollen grains, the animal dander, right? Majority of these things are usually seen in the environment. This is what is happening. Now, how does the immune system respond to these particular things, right? So let's quickly take up how our immune system would respond to these sort of allergens, right? So whenever an allergen enters into the body, right? Whenever an allergen enters into the body, say this is an allergen here, which is the antigen. Whenever the allergen enters into the body, again the same thing. The here actually the type of immune response that is brought about here is mostly the humoral immunity, right? It is mostly the humoral immunity. What exactly happens is whenever an allergen enters into the body, first the B cells would get activated, right? The B cells would get activated. So this comes across or in contact with the B cells. Now we know what the B cells do upon activation. So B cells will start synthesizing two types of cells, the memory cells, and the other one is called the plasma cells or the effector cells. Now we know what these plasma cells do. These plasma cells would start synthesizing the antibodies, right? One important thing here, the type of antibody that is synthesized against the allergen, right? The type of antibody which is synthesized against the allergen is this IgE type of antibody. This is an important one, what you need to remember, right? IgE type of antibody is being synthesized against this particular allergen that has entered into the body. Now, what does this IgE do? IgE does not go and directly target the antigen, right? What does IgE do? If you could recollect when we were talking about the classes of antibodies, when we saw about the function of IgE, I told you that IgE are usually seen as membrane-bound antibodies, right? They're usually seen as membrane-bound antibodies on what? On two types of cells I told you. One is called the mast cells and the other one is called basophils. Right. So as and when IgE antibody is synthesized, those IgE antibodies will go and get deposited on two types of cells. They would go and get deposited on two types of cells. One is called the mast cells, right? One is called the mast cells. Other one is the basophils, right? So they would go and get deposited in two types of cells, mast cells and basophils. Now this mast cells and basophils, they have receptors on their surface, right? So they have receptors on their surface. So these receptors are usually considered as FC receptors, right? So they have FC receptors for IgE class of antibody. So if you could recollect, when we were talking about the structure of antibody, we said that antibody structure could be divided into two parts or two regions. One was called Fab fragment, fragment of antigen binding. Second one is the FC fragment, the stop part, right? So this mast cells and basophils, they have receptors for FC fragment, right? They have FC receptors for IgE type of antigen. So as and when IgE antibodies are synthesized, those IgE antibodies will now come and bind to the FC receptors which are present on the basophils and the mast cells. This is how they come and bind. So this is our IgE type of antibody. Right. So once IgE antibody binds to the FC receptors on the basophils and mast cells, this mast cells and basophils will now undergo the process of activation. Right, basophils and mast cells will now undergo activation. Once they undergo activation, they would start releasing certain chemical substances. Right, so they would start releasing certain chemical substances. So what are the chemical substances released by basophils and mast cells? We have already studied about this in our last year, right? The basophils and mast cells, they release three major types of chemical substances. One is called histamine, histamine, second one is called serotonin, and the third one is called bradykinin. Right? So these are the chemical substances which are released by the basophils and mast cells. So when are these chemical substances actually released? Whenever the IgE antibodies come and bind to the Fc receptors present on the basophils and mast cells. So once they bind to the Fc receptors present on the basophils and mast cells, the basophils and mast cells will now undergo the process of activation. Upon activation, they would start releasing these three chemical substances. Now, these three chemical substances will now start targeting the antigen that has or the allergen that has entered inside the body. Right. Now, how does they actually do it? Along with targeting the antigen, this histamine, whatever is there, right? So the histamine, whatever is being released, that histamine would bring about swelling. We call it as edema, right? It brings about edema, which is nothing but swelling of that particular tissue or that particular part. Second one, it brings about a condition called as erythema. Erythema is nothing but redness. So that particular part, it becomes reddish in color. And along with that, it also leads to some sort of inflammatory reactions. Right? It would lead to some sort of inflammatory reactions present in the body. This is how the immune system actually responds to the allergic substance or allergen that has entered into the body. So what exactly is happening? Right? So quickly going through it once again. Whenever an allergen enters into the body, it could be any of the allergen, either the dust particle or pollen grain or animal dander or fruit, vegetable or any drug, whatever it is. Whenever an allergen enters into the body, the B cells would first get activated. Upon activation, the B cells will now start undergoing differentiation to produce two types of cells, memory cells and the plasma cells. The plasma cells will now start synthesizing the antibodies. The type of antibody which is synthesized against the allergen is this IgE type of antibody, IgE class of antibody, immunoglobulin E. Now, this IgE type of antibody is always a membrane-bound antibody. They have their Fc receptors present on the membranes of mast cells and basophils. Right. So, as and when IgE antibodies are released, that IgE will go and bind to the Fc receptors that is present on mast cells and basophils. Once they bind to the Fc receptors on mast cells and basophils, these mast cells and basophils will become activated. Once they get activated, they would now start releasing certain chemical substances such as histamine, serotonin, and bradykinin. All of these chemical substances will go and start targeting the antigen, and it would bring about the complete destruction of the antigen. Now, along with destroying the antigen, what exactly these again does is, especially histamine, they go and start bringing about some sort of swelling in the surrounding tissue. We call it as edema. It brings about redness of that particular tissue. We call it as erythema, and also it leads to some sort of inflammatory reactions in the body. Right? So this is what is happening. Along with targeting the specific antigen that has entered into the body, whatever cells and tissues are there surrounding that antigen, those human tissues, self tissues or self cells are also being targeted by these particular substances. So, whenever a person is suffering from this, what the doctors usually do is to reduce the process of allergic reaction or to reduce the inflammatory reaction, the doctors would usually give or administer something called as antihistamines. Along with that, they would also administer something called as adrenaline. So, we are all familiar with adrenaline, and also some of the steroids. Right? So, administration with antihistamines, adrenaline, as well as steroids will quickly start reducing the allergic reactions in the body. If it is not treated, it would start damaging the nearby cells also, bringing about the destruction of our own cells. That's the reason why we start calling it as exaggerated immune response. Along with destroying the antigen that has entered into the body, even the self cells the normal cells are also being destroyed because of the release of histamine, serotonin, and bradykinin in a very, very high concentration. To avoid this or to overcome this or to reduce this allergic reaction, the patient should be administered with either antihistamines, adrenaline, or some of the steroids. Right. So this is what we have. Right. So what most of the doctors actually believe is the modern lifestyle. Right. So the modern day lifestyle is one of the major cause for reduced immunity and becoming more sensitive to certain substances such as these. Because especially in case of major cities, most of the individuals or children, they are usually seen in the indoor zone. They usually don't come out. They are usually seen indoors. Since they are seen indoors, they are less what we can say, they are less exposed to some of the antigens that are present in the environment. Over a period of time, they would become highly sensitive to these particular antigens, which would lead to this condition called as hypersensitivity. Right. So this is the first type of immune disorder what we have, which is called as hypersensitivity or allergy, which is nothing but the exaggerated immune response of the body, right, against a particular pathogen present in the environment. So how does it bring about the immune response? It is mostly mediated by IgE type of antibody. So whenever we talk about allergic reaction, automatically three things should come into your mind. One is IgE type of antibodies involved. Along with that, mast cells and basophils are actually involved. Right, so this is what we have with the first type of immune disorder, which is called as hypersensitivity. Right. So in the next session, let us take up with two more other type of disorders. One is called autoimmunity, and the other one is called the immunodeficiency. So we'll take up with those two disorders in the next session. Right.